Hello, I'm Dr. Robin Warner. I just recently graduated from the Hospital for Special Surgery Neuromuscular Medicine program, and this is my lecture on observing patterns in MRI with QSM in patients with SOD1 genetic ALS. I have no disclosures. So in this lecture, I will introduce you to SOD and the SOD1 gene, introduce a proposed cellular mechanism that I came up with after reviewing some literature, and explain what QSM is in the context of MRI. Then we'll do an overview of our study and discuss the findings. Only about 10% of ALS cases are genetic, 90% are sporadic. Most of the genetic cases are autosomal dominant, but they can also be X-linked or recessive. Several genes can cause ALS. Most of these genes are not actually causal, but are disease-modifying genes. C9orf72, which is the most common gene, is causal. We'll be discussing the second most common gene, SOD1, which occurs in 20% of patients. So the SOD1 gene is located on the 21st chromosome, and it contains 5 exons. 150 mutations of these exons have been identified. Most of them are missense mutations, but they can also be nonsense mutations or insertions or deletions. So we know that there is a complex combination of molecular pathways that actually cause ALS. These include oxidative stress, aberrant RNA processing, misfolding of proteins, aggregation of insoluble proteins, all together in some combination cause ALS. For basic understanding, I compiled this oversimplified mechanism. So microglia secrete cytokines, which stimulates inflammation, recruiting astrocytes, and the astrocytes release nitric oxide and prostaglandin E2. So far, this is all normal functioning of the cells. The SOD comes to convert nitric oxide to peroxide and oxygen, which is what it does. However, in ALS, this SOD is mutated, and instead of doing its job, it aggregates. This leads to stress on the endoplasmic reticulum as it tries to clean up after all of the aggregation. The endoplasmic reticulum also traffics endosomes, and this can cause disruption in the function of organelles. This includes the mitochondrial disruption, which activates apoptosis of the neuron. Endoplasmic reticular stress also causes dysfunction in axonal transport. Dysfunctional axons die back as a part of cell function, and so this leads to axonal loss in ALS patients. As you can see, there are many different mechanisms that all contribute to motor neuron degeneration in ALS. And each one of the mutations involved come in at a different point or multiple points in these mechanisms. So what is SOD? It's short for a superoxide dismutase. This is a cytoplasmic enzyme that has a functional component at the copper ion and a structural component at a zinc ion. It's 153 amino acids long. Its job is to convert oxygen radicals, such as nitric oxide, to peroxide and oxygen. Catalase, which pre-existed in the cell, it's more ancient, then takes the peroxide and turns it into water and oxygen, the waste products of the cell. This is an extremely stable protein as a dimer, but it can unfold to a folded monomer intermediate, which is unstable, and then move on to two unfolded monomers, which are also stable, but tend to aggregate. So as you can see, we have the dimers on the left, the monomers on the right. The intermediate is not included in this diagram. The top is a healthy SOD molecule from a wild-type SOD1 gene. The bottom is a mutated SOD1 A4V molecule, which is more stable as a monomer. The monomer tends to aggregate. The top one is more stable as the dimer, which is more functional and does not aggregate. So this is a diagram of all the different locations on the dimer on the left, the monomer on the right, turn 90 degrees, of the SOD1 mutations that occur in ALS. You can see the locations are highlighted in yellow. So on pathological tissue sections of the central nervous system, you can see certain areas are more affected than others in SOD1 ALS. Specifically, the spinal anterior horn have weakly ubiquitin positive, TDP43 negative, SOD1 negative, neurofilament positive, intracytoplasmic hyaline conglomerates. The motor cortex also has these conglomerates, but it's more infrequent than in the spinal anterior horn. And frontal and temporal regions may also contain these conglomerates, but even more infrequently than in the motor cortex. In the medulla, you can see changes in the Clark's nucleus, dorsal horn, nucleus ambiguus, and Onuf's nucleus. There will also be some axonal generation in the descending axons. Here are some pathological sections of a patient who has SOD1 ALS, specifically the A4V mutation. The left and center are of spinal motor neurons, and the right is in the motor cortex. The left shows a Lewy body-like inclusion in the spinal motor neuron, and then in the center you can see this is accompanied by misfolded SOD1. On the right, there is an absence of misfolded SOD1 in the motor cortex. So what is MRI with QSM? QSM stands for Quantitative Susceptibility Mapping. This is an analysis that identifies degenerating neurons in the motor cortex due to abnormal iron deposition. It can also be applied to other areas of the brain, such as the basal ganglia. Currently, in studying ALS patients, we use this method to examine the motor cortex, which typically has increased iron due to dying cells. When a cell dies, iron is left behind, and the brain is unable to clear out all of this iron, so some of it is left behind. The amount of iron deposition correlates with the clinical syndrome and becomes more prominent as the patient worsens. So for example, we can see in a patient who has onset of ALS in a flail arm, the motor cortex that correlates with the weakness will have increased susceptibility. And then as that patient becomes worse, the susceptibility will become more and more increased with time. And this is what it looks like. So here's a brain section with the hook of the hand. Both sides have increased susceptibility and it's brighter. Sometimes it's darker on other scans. Ours shows up as a brightness. It's very similar to SWI. However, we aim to one day quantify the susceptibility with this particular sequence. In our study, we retrospectively assessed the QSM sequence of the MRIs in ALS patients with SOD1 mutations. And we wanted to see if there was any specific pattern to their accumulation of iron as opposed to other kinds of ALS, sporadic or other genetic. This was a retrospective study, like I said. The patients were collected between 2013 and 2018 from the Hospitals for Special Surgery's neurology department, seen with ALS, having an SOD1 mutation that was confirmed. We identified 7 patients, but only 3 of them had the MRI with QSM sequencing. So we sat with the neuroradiologist and examined these scans. And we found no distinguishing abnormalities in the brain MRIs of any of the patients who had SOD1 genetic ALS. So you see the 3 patients, 2 of which had A4V mutations, and those were normal MRIs. And then the 3rd patient had a D90 mutation, and there was some corticospinal tract enhancement. Those findings were supportive of another study that had been done in ALS A4V patients that showed that this subtype, which is the most common subtype, does not affect the corticospinal tracts. We think that the fact that we cannot reflect the amount of disease burden may be due to the fact that maybe protein aggregation and cell death are not directly related to each other, or that the aggregates can affect certain neurons or tracts more than others that we're not looking at. The corticospinal tracts were affected in the SOD1 D90 subtype, and so therefore MRI with QSM may not be useful in distinguishing ALS due to SOD1 mutation from any other kind of ALS patient. These are my references. Here is my abstract for review. Thank you to everybody who came and stopped by my poster earlier this week. And for more information, you can download for free my chapter on ALS. This is the link and the DOI number. If you have any questions or comments, please feel free to contact me at robin.patrizzo@gmail.com. Thank you very much.